This video is my second video talking about mutation to genetics students. The first video goes through the causes and effects of mutation. This one will begin with how mutations are repaired, but then go through a diverse series of topics, finishing up the topic of mutation. So this is a problem when ultraviolet light strikes two adjacent thymines and causes them to stick together, causing something known as a thymine dimer, because when the cell goes to replicate the next time, it will then interpret those two nucleotides as one single one, and that will cause a mutation. Other organisms face similar problems. And so some organisms have specific enzymes like photolyase, which will now then break up this dimer as a part of the process photoreactivation. Okay, so humans don't have that, but we can fix something like this through a second mechanism known as excision repair. So first I'd like to make the point that all organisms have to repair mutations and that there are different mechanisms for doing so, and not all organisms have the same mechanism. So here was an example of one that was not found in humans. So there are a number of different ways that this can then be performed, because obviously here's a distortion and enzymes which are following the DNA molecule, and there are some enzymes that's what they do. They will notice that there's an abnormal shape here, a distortion, and then they will then excise something. Now there are different enzymes here, so a glycosylase might just excise the nitrogenous base and leave the sugar phosphate background. There are other enzymes which can make nicks ahead and behind the distortion and then cut that area out. So the distorted area has been removed. Okay, so once again, there are different ways of doing this. You could just take out nitrogenous bases, not even the whole nucleotide. You can remove, entire nucleotides, etc. So what then happens is DNA polymerase then can add new nucleotides in the 5' to 3' direction, just as it would during DNA replication. And then, although, I just noticed that these are going the wrong way. Sorry about that. And then an enzyme called DNA ligase can then seal the nick. And so therefore, what was a distorted region has now been fixed. Now, for some reason, some of the excision repair in eukaryotes, such as humans, is tied to transcription. So if an area is going to be transcribed, used, so genes are going to be activated, then DNA that is distorted is fixed. Why is this linked to transcription? Not sure. Is this just an energy conserving mechanism that you would not bother fixing an area that you're not using and that you would simply repair those areas which are about to be used, or just making sure that before you activate an area that it is fixed? And so there are genes such as BRCA1 and xeroderma pigmentosum where they encode proteins which are involved in this excision repair tied to transcription. Now, some may associate BRCA1 with breast cancer. BRCA1, and some refer to it as the breast cancer gene. Obviously, we don't have genes which cause breast cancer, but we do have genes which try to stop breast cancer. So BRCA1 has multiple roles, one of which is functioning in excision repair. And if BRCA1 is mutated, then it might not be able to perform that. And that might make an individual susceptible then to cancer. There is another form of DNA repair known as mismatch repair, and about 99% of our mutation repair is actually mismatch repair. Now, one of the times that we're vulnerable to make mistakes is that when we are replicating our DNA, so making a double-stranded molecule, single-stranded, and then filling in complementary base pairs. DNA polymerase makes mistakes, but then it also fixes mistakes. It has its own proofreading ability. And so after DNA polymerase fixes its own mistakes, the resulting error rate is about 1 mistake per million nucleotides. Now, that's good, but that is still too many. So notice here that the yellow guanine is bound to the blue thymine. That's an incorrect base pairing, and that would then distort a little bit. The nucleotides wouldn't fit together as well as they should. And so that's when this mismatch repair would then excise the nucleotides along around the distortion. And now DNA polymerase would then add in nucleotides going 5' to 3'. Once again, in that one, they were going in the correct direction. Sorry for the previous mistake. And thus the error, it is fixed. And once again, mismatch repair is tied to DNA replication. So following the replication fork, the cell would then look for distortions following a problem with replication. Now, how would the cell, if it recognizes there's a problem here, there's a mismatch, which do you fix? Because if one nucleotide's the correct one and one is the incorrect one, how would you know, for example, that in the previous example, that it was the yellow guanine? Which of these should be removed? Should be the guanine be kept and that be changed to a cytosine, or should the thymine be kept and that changed to an adenine? Well, you may recall that the cytosine nucleotides here in green can be methylated. A methyl group, a CH3, can be added to them. And that after cells divide, that the new strand would then have to later have methyl groups added to it. So its methylation matched that of the parent strand. But prior to that happening, this top strand would be methylated because that was the original strand. And this was the new one that was synthesized to be complementary to it. It would not be methylated. And so if a mismatch were found here, then the enzymes would recognize that if this is the methylated strand up top, that would be kept, and it would then be the guanine which would be removed and replaced. Now, that's apparently a reason, maybe the major reason, that while DNA uses thymine, RNA uses uracil, because if DNA used uracil, then there could be a fairly common change where this could become a cytosine, and this would affect the methylation in a way that would make it hard for cytosine could become uracil and thus would not be methylated, and thus you would have a lesser ability to perform mismatch repair. So this is important. 99% of our mutation repair is this, and once again, that may be the reason that we don't use uracil in DNA, because then cytosines, which became uracils, you couldn't distinguish between the original strand and the following strand. The most complex of the mutation repairs would be post-replicative repair. So imagine that there's a double-stranded molecule, it separates, and now each of the two single strands then has DNA, new complementary bases attached to it. Well, here, notice that one of the original strands, so there was an original strand, say, up top, and then a new strand was synthesized complementary to it. If the same thing happens here, here's the original strand, but the original strand has a problem. We want to make a new strand, but you can't make a new strand if the original strand has a problem. What to do? Well, in bacteria, there's a protein called REC-A, and humans have homologs to it, such as a DMC and RAD, as you'll see. And what this does, and it's complicated, the proteins, and they can form a complex, can bind the damaged region and then migrate it to the good strand. So notice that this single-stranded area had no distortions, and so therefore it's already been replicated. And so Rec-A transports the damaged region to the other formerly matching strand. And it searches for homologous region. Where is a matching sequence? When it finds a matching sequence, it will cut one of the two good molecules. So once again, there was a good strand, an undamaged strand, that was then replicated. So now on the top strand, there are two correct sequences. Rec-A will cut one of them and then replace that newly cut one with the damaged strand. So it's going to displace part of the correct sequence onto the damaged strand. So now there's a good copy on both of the two strands, and then later DNA polymerase will then add new nucleotides, and ligase will then repair them. So it cut away the damaged area where there was a thymine dimer. It cut this good sequence and then pasted it here, replacing the damaged sequence, so that now each of these two sequences have a correct undamaged area, and now complementary bases can be added. So post-replicative repair involves a protein Rec-A or its homologs actually binding to the damaged area and then searching for a homologous region. Okay, so that's complicated, but it's also so very exciting because if you've studied meiosis, and obviously, you know, probably at some point in your biology you have, you might think, wait a minute, homologous regions being cut and pasted, that reminds me of the crossing over, which occurs during meiosis in preparation for sexual reproduction. And it uses these same proteins. So here's the fascinating thing. Rec-A evolved in bacteria to repair mutations. But once bacteria and ancestral organisms had a mechanism to seek out homologous sequences and to cut and paste, eukaryotes, which invented sexual reproduction, took advantage of what was originally a mutation repair and then now used it to ensure homologous chromosomes. So the challenge for an organism preparing to make gametes for sexual reproduction is, how do you make sure that a gamete has the right chromosomes? I mean, if you're cutting, you know, if you're splitting piles of chromosomes in half, how do you make sure that, you know, the haploid gamete has all the right chromosomes? Well, this way. So this is what happens during meiosis. Double-stranded breaks are introduced. So whether it be us or other sexually reproducing organisms, they deliberately damage the DNA. They deliberately make breaks in what was a perfect DNA sequence. So now there's a problem. There's a hole that has to be fixed. Why? Because we put it there. All right, we broke our DNA. We made mistakes. And in order to fix the mistakes, the homologs of Rec-A are going to bind the other strand and search for the homologous region which matches that. So we initiate these double-stranded breaks on purpose, right? In yeast, maybe 160 times, but 400 times in mice. And these double-stranded breaks then become the sites where crossing over occurs. Now, not every double-stranded break gets fixed involving crossing over, but this is where crossing over occurs. And I can't emphasize this enough. Not only does this happen during meiosis and precursor to sexual reproduction, it has to happen. If chromosomes don't do this, if a cell doesn't do this, then the cell will fail and not produce gametes. It will die. So we destroy cells that don't do this crossing over, because without which there's no guarantee that you're sorting the chromosomes correctly into opposing piles. And so it is homologs like DMC1 RAD51 and RAD51. These are, eukaryotic and human versions of REC-A. They then search for matching regions in order to fix the double-stranded break switch we made. But as a result, you've just guaranteed that these two chromosomes have homologous regions. They match, all right? You've just assured that. And so what evolved as a way of repairing mutations, and we do use it as a form of mutation repair. In addition, it then is required for sexual reproduction. So a spermatocyte or an oocyte in pachytene and diploatine of meiosis, which does not do this, it does not then ever produce gametes. It will then be destroyed. And so this is then required. And when we talk then about a chromosome now being mostly from, say the male parent, but then having some of the female parent's genes, this was how this was managed. So this is not only critical for mutation repair, but then also for sexual reproduction. Okay. Now I'd like to just kind of mention a couple of topics wrapping up on mutation. So the major tumor suppressors like P53 and BRCA. P53 is perhaps the major protector of the genome to prevent from cancer. All right, so if we prevent cancer in our lives, P53 is more so than any other protein the reason for it, and it is mutated in more cancers than any other mistake. So not having a functional P53 protein is more likely to cause cancer than anything else. This and what BRCA1 that I mentioned previously and other tumor suppressors too, is they are involved in stopping cells dividing after they've had mutations. And so DNA can be damaged, mutations can be induced, and we have proteins which before a cell can progress through the cell cycle, we're checking this, and damage must be repaired. All right, so P53 can have two roles. One, it can recognize damage and then help to signal the DNA repair mechanisms. But if failing that, if the damage isn't repaired, then it can help trigger the programmed cell death, the apoptosis. So we try to fix our DNA, but if a cell's DNA mistakes aren't fixed, then we kill that cell. We don't want cells dividing if they have mistakes. So tumor repair is not only important, but when we get back to the cell cycle, there are checkpoints. And unless we have passed then these points of tumor repair, then the cell would not be allowed to progress. Now, just because once again, this is kind of the end of the chapter that on mutation, I then have a couple of final topics just to tie in. The next topic I'd like to go into is what's called the complementation test. So let's say here we have a stock of white-eyed flies that say comes from lab number 1. So let's call this strain number 1. And we made it to red-eyed flies, and we can do a red-eyed female to a white-eyed male, and the reverse, and then observe. And in this strain, we'll notice that it's recessive because all of the flies then have red eyes. Great. So that's what we see in strain number 1. Now let's say we go to a different lab and they have white-eyed flies as well. And we ask the question, is it in the same gene? All right, or are there multiple genes which can cause these white-eyed flies? So once again, we mate the red-eyed true breeding strain to the white-eyed true breeding strain, and we notice that it's recessive. So notice if the male parent was the one with white eyes, then all of the offspring have red eyes. But if the female parent had white eyes, notice that their male offspring have white eyes, that this is recessive, but it's sex-linked. So clearly this is a different gene than the first strain. So there are now two different genes causing the same phenotype. So strain 2, all right, where the mutation is sex-linked, is different from strain 1. Now, we have just shown this because one is sex-linked and one isn't. But let's say that one wasn't sex-linked. How would you know? Let's say you have two white-eyed flies. How would you know if they're mutations in the same gene or different genes? Well, what you could do is the complementation test, where you could mate a female from strain 1 to a male of strain 2 and vice versa. So now you have heterozygotes. And then the question then is, do they have red eyes? All right. Well, here in this case, the female of strain 2 has white-eyed offspring because that's sex-linked, but we knew that. So here, the key is look at the red-eyed flies. If the white-eyed mutations were in the same gene, all of the flies would have white eyes, all right? But they're not. So what we know is that there are multiple conditions where you could have white eyes in a fruit fly. So here, this fly, this is the female of strain 2. She's homozygous for a mutation in the gene white. That's why she has white eyes. And when she passes that on to her male offspring, that's why she has, it would have then white eyes. The male from strain 2, notice that it has one white mutation on the X chromosome, and the Y chromosome doesn't have a white gene. But there is another way to have white eyes in flies. You could be simultaneously homozygous recessive for mutations in brown and scarlet. So this fly and the corresponding male, they have white eyes not because of mutations in the white eye gene, but because they have mutations in the brown and scarlet genes, all right? So why would white-eyed flies crossed with white-eyed flies give you red-eyed flies? Why does that make sense? Well, the answer is, and if you can look at the bottom, sorry for the play bar here, notice that there are 3 genes we're looking at: brown, scarlet, and white. Now, notice in this cross, this parent is homozygous for the wild type alleles for brown and scarlet. So these offspring will get wild type alleles for brown and scarlet because one parent, that's all they had to give. Their mutation was in the white gene. Whereas the other parent has wild type alleles for the white gene, their mutations were in brown and scarlet. Either way, this fly is not homozygous for any of these genes, brown, scarlet, or I'm sorry, any of these mutations, brown, scarlet, or white, because it gets a wild type allele for all 3 of them. So this is a heterozygous now for those 3 genes. This fly is heterozygous for those 3 genes, as would this fly. And because all these mutations are recessive, none of these flies would have white eyes. The point is, we started off earlier that we knew because one was sex-linked and one wasn't, that we were talking about different genes here. But if you didn't know that, how would you know that the strain 1 from this lab is a mutation in the same gene as the strain 2 from this other lab? You would do a complementation test. If heterozygotes between the 2 strains have the wild type phenotype, these are not alleles of the same gene, all right? And so therefore they would be in different genes. And so the complementation test, we've pointed out that these are not alleles of the same gene. They are non-allelic, okay? Now, if we did observe that when you cross these strains, that the heterozygotes between the 2 strains would have the mutant phenotype, we would say, oh, these 2 strains have mutations in the same gene causing white eyes. They are alleles. They are functional alleles of each other in the same gene. So complementation tests, we can show whether 2 different mutations are in the same gene or in different genes. Next question. So lab number 1 and lab number 2 have mutations causing white eyes. I'm sorry, lab number 2 and lab number 3. Let's say we have strains causing white eyes. And We know that it's the same gene. We've shown through the complementation test that it's the same gene. Great. Is it in the same spot of that gene? Okay, because a gene is a big thing, right? I mean, you can have hundreds, thousands of amino acids coded for, and especially when you consider the introns, the genes can now be much longer than that. So there could be a difference between, say, the beginning of a gene and the end of a gene. So if you can have two mutations in the same gene, are they in the same spot or are they in different spots? All right. Well, make your heterozygotes. All right. And now then the question is, do you rarely observe a red-eyed fly? All right. If you do, how could that be explained? And the way that you could explain it is that these are structural alleles. So they're alleles of the same gene, but they are structural alleles at different spots in the same gene, but at different spots. And let me show you how that would work. So let's say you create a heterozygote with, say, the X chromosome from one parent and that you get from the male. So here's an X chromosome from the female parent of this fly, and here's an X chromosome from the male parent of this fly. Okay. If they are in different spots, what could happen? Now, crossing over doesn't, it always happens, but it doesn't always happen in any specific spot, much less in the same gene. But if it does occur in the middle of this gene, so if we swap these sections of DNA, if cuts are made, and we move this little blue section over here, this little red section over here, now one of the two X chromosomes would have both mutations, one, and that would give a white-eyed fly. Whereas now one of the X chromosomes doesn't have any mutations, so it only has wild type sequence in these two sites. And so it would result in a red-eyed fly. So when we cross strains 2 and 3, if we're observing that there are occasionally these red-eyed flies, then that would indicate that the two mutations are in the same gene, but not at the same site of the gene, because we're getting recombination occasionally separating them, giving us a wild type fly. If you never observe these red-eyed flies, if all you have are white-eyed flies, then you might conclude that the mutations in the same in these two strains are located at the exact same spot in the gene. Okay. Now, I just want to mention this other thing at this point. We're getting to the point where you would stop looking at flies because the number of flies you would have to screen is just far too much. Now you would start looking at bacteria and viruses. But let's say you don't cross strain 2 and 3. Let's say that you just have strain 2. Strain 2 is all by itself in a vial, continually crossing with it itself, but that every now and then you have a rare red-eyed fly. Now this would be much rarer than the previous event. All right, so there are actually two events that could cause red-eyed flies, crossing over between two X chromosomes that have structural alleles. That's rare. But what I'm about to say is even rarer. That let's say there was a mutation that inactivated this gene so that, I don't know, so some sort of a codon that was supposed to be AGA and code for an amino acid now codes for a UGA, which is a stop codon. The transcript doesn't make a long enough protein, it doesn't work, so there's white eyes. So there's a point mutation, one nucleotide was changed. Well, you could have a second mutation where the UGA mutates back to the AGA. All right, so it would revert to the original sequence. Now that would have to be that exact nucleotide would have to mutate, and it would have to mutate back to the original. So this is an extremely rare event, but nevertheless, this reversion mutation could then give us a red-eyed flies again. Now, if you're going to study mutations, these are types of questions you might ask. But we're once again getting into where you would no longer be looking at flies because these are so rare events. You just have better things to do than to count flies all day long. Why would you do this? Well, because there's different types of mutations, and you might want to distinguish between the different types. So for example, a point mutation, there would be the chance of that reverting to the original condition. If, however, it was a deletion mutation, so the mutation was actually that now you're missing a large portion of the gene, there's no way that a subsequent mutation reverts that to the original sequence. So this would never give you a red-eyed fly. And so here you could distinguish between a point mutation, which would occasionally give you reversion mutations with white eyes, with red eyes, and a deletion mutation, which would never give you a reversion mutation with red eyes. But once again, no one wants to count 1 million fruit flies. But if you're looking at bacteria, all right, you can then easily screen millions, billions of bacteria. And so there are, different types of mutations in bacteria. So you can have prototrophs, which can make their own nutrients, or auxotrophs, which are mutated versions of the prototrophs. So now one of their genes doesn't work. All right, they can't make a nutrient, and therefore you have to give it to them in their medium, or else they die. And then you could then ask, what is the frequency with which an auxotroph then mutates back and becomes a prototroph? So that's a type of bacteria you could screen in, as a type of mutation you could screen in bacteria. And then there are strains of bacteria which don't allow viral infections, strains which do, and you could notice at which point a bacterial strain would mutate and have the other phenotype. Then you can even talk about mutations in viruses. There are wild type viruses which can infect both E. coli strain K and E. coli strain B. But then there's mutant T4s which can infect E. coli strain B, but not E. coli strain K. All right, so there's mutant virus and wild type virus. But sometimes a mutant virus might have a reversion mutation going back to the wild type condition. So something that would start off unable to infect E. coli K could then revert back to the original condition where now it could infect E. coli K. So you could actually measure the frequency of these reversion mutations. But once again, while a point mutation could perhaps mutate back. All right, so notice that it started off as a cytosine that was a mutant that didn't work as well. You could measure what's the frequency that it goes back to the original thymine. A deletion mutation couldn't do that. You couldn't have a subsequent mutation, replace this sequence. All right, so there are geneticists who study mutations, but after a certain point, sometimes you are studying then viruses and bacteria. Because, for example, if you're looking for what are the odds of getting, recombination within a single gene, or, not just between genes like you could like in fruit flies, but within a gene, all right, recombination between this nucleotide and that nucleotide in the same gene, or a reversion mutation of this nucleotide back to the wild type nucleotide, but a mutation only at that spot. Well, these are such rare events that probably you're going to be working with things like bacteria or viruses. Other mutation experiments which use bacteria and viruses would be things like, is something a carcinogen? Well, obviously you could test it on animals, but before you got to that, you could test it on bacteria, because you could measure the mutation rate in bacteria. At what point, at what frequency do these bacteria mutate from this phenotype to that phenotype? And once again, there's different phenotypes in bacteria. After I've exposed it to this new chemical that I'm thinking of putting into food as an additive, does that change the mutation rate of bacteria? Does it become now more common? And so a lot of people, are studying mutations, mutations rate, even in bacteria and viruses. But this is a way of screening out carcinogens and making our food safer and our products safer. Now, this is kind of silly. This video was talking about why are there so many species on Earth? What is the source of the variation? And the variation comes from, mutation and the random assortment of alleles in sexual reproduction. So when we look at the planet, we don't just have one type of sparrow, one type of frog, one type of monkey, one type of organism. You've got lots of kinds of dog or cat or monkey, etc. So mutations are ultimately the major source of variation, and then sexual recombination can jumble these. So while we speak of mutation as being a negative thing, because obviously if we were to get mutations, then that could predispose us to a disease, but nevertheless mutations are a source of variation, and ultimately the raw material through which adaptation and evolution works. Without mutation, life would not have changed. All right, life was once just bacteria. The reason it's not just bacteria anymore is because of change over time. Another point that I made before, but I've made again, very often mutations in multiple genes can cause the same phenotype, not just white eyes in fruit flies. But some people who suffer liver cancer have a mutation in P53. But some people who have liver cancer have a mutation in the insulin-like growth factor 2 receptor. Some people have liver cancer caused by other mutations, such as this one in a second messenger system, etc. So there are lots of different genes whose mutations can cause heart disease or asthma or cancer, etc. And so, that principle I mentioned earlier is, actually something that one can apply then to people. This next question is a bit historical. What is the cause of mutation? And do mutations just happen, or are they always induced? Well, when we talk about mutations and the scientific study of mutations, a good place to start is in the 1940s. Luria and Delbruck, they performed an experiment to answer that question. And what they did is they started off with a bacterial strain susceptible to a specific virus, but which can undergo mutations to become resistant to that virus. And so I'm just kind of simulating their experiment in two different examples, this one and that one. But so here's a bacterium, and they can recognize the phenotype of the mutant bacteria, okay? And then they added a virus, all right? And so the mutation will be resistant to this virus, or at least those are the mutants that are going to live on these plates, because if the bacteria aren't mutant, then the virus will kill them and they'll die. So if there are mutants which are resistant to the virus, they'll grow on these plates, otherwise they die. Now, the idea was that if it is the virus addition which is the stimulus for the mutation. So if you ask these bacteria, why did you mutate? If the answer is, well, because you added virus, and the addition of the virus then induced the mutation, if that's why the bacteria then mutated, then because you did it in 5 different tubes at the same exact way, then they should all undergo mutation at same levels. And so if you then take those 5 different tubes and put the contents on 5 different plates, then whatever the frequency of mutation is in one should be about the same as the other, because the virus which was inducing the mutation, then that was added to all of them. But if it's not the addition of the virus, if mutation just occurs randomly, then once you made this bacterial culture, maybe in this culture, the mutation happened early, all right? And let's say you started adding viruses, whatever. But then a mutation occurred here. And then a mutation occurred in another tube or another tube. So it's not the addition of the virus, it's just something that happens at random over time. Sometimes earlier, sometimes later, and sometimes maybe not at all, as in tube 3 here. Well, if that's then the case, then whichever tube had the mutation occur the earliest, tube 2 in this case, then would have more mutant bacteria, because as the bacteria divide, if it occurred very early in the culture, then more progeny will then have the mutation. There will be more mutant bacteria in the plate resulting from this one than this one. And this tube, the mutation came later. So they had this hypothesis, that we would tell the difference based on the number of mutations. So this is what they observed. All right. Some plates would have a lot of mutants. Some had less, some had less, some had none. That's consistent with the idea that the mutations are developing at random and not being induced by viruses, because if it were the opposite, then they would notice the following, where all 5 of the plates would have about the same frequency of mutation. In this video, just because for a lot of my students, I think hearing this twice would benefit, I just say, all right, well, let's imagine we do the same thing where we have bacteria that are mutant in that they can't make their own metabolites. But now we're going to add a medium for them to grow in, but it's missing a metabolite. So we're now looking for which ones now have a second mutation back to the original prototrophic condition where they can make their own media. So if you add the minimal media, which means now they're either going to have to make their own food or they die, because the media that we're adding doesn't have the metabolite that they are unable to make. This, then we can now screen for mutations back to the original condition where they can make their own food, because only the bacteria which make their own food would be able to grow here. If it was the addition of the minimal media, which was the stimulus for the mutations to occur, then we should have similar levels of growth on all of these plates. If, however, those back mutations occurred randomly, all right, so let's say that in this one, it occurs first in, I just forget which tube goes first. All right, so in this tube, say tube 5 goes first, tube 2 goes second. So if this occurred earliest, then this tube should have the most mutants, this one have the second most, etc. And that would be consistent with mutations occurring at random. And so that's what Luria and Delbruck observed, once again, kind of simulating, their experiments. But they showed that, look, because the frequency of mutation varies, some more, some less, some none at all, it does not look like this. So this leads us to believe that mutations are just constantly occurring. The cells don't know that the mutation that has occurred is going to help them survive. It's not like they get a virus and think, oh, I need to mutate a resistance to this virus. Everybody tried. That's not the way it happens. Mutations, simply occur, and then it's just simply dumb luck whether or not an organism would then have a mutation which helps it survive. Finally, just a quick note. This actually would have been better in the previous video that I had made, but I've just added this at this video. And so in the previous video, I talked about what a frameshift mutation was. Let me first talk about letters in a sentence. So if there was a sentence, the big dog sat on a red car, all right? If I were to look for 3-letter words here, all right, then I break it into units, and that kind of makes sense. If I were to change one spot, do what's called a point mutation, if I were to talk about, say, then DNA, then here with that point mutation, the THE is changed to TRE. So that changes the meaning of one word. So clearly that affects the sentence, but maybe you can still read the sentence and get close enough an idea of what that a sentence would mean. But adding or deleting letters, that then is different. So notice that if I add letters, all right, so let's say I add the letter X, not only does it change this first word, but now the E, which was part of word 1, is now part of word 2. And if I were to read it looking for 3-letter words, I would now read thx, ebi, gado, gasa, tan, ar, daka. So that didn't just affect one word, it affected all of the subsequent words downstream of that. It altered the reading frame. And so therefore that's a much more serious effect than the point mutation, which occurred first. And the same thing would result if I deleted a letter or two. Now, this would affect if I add or delete a letter or two. If I add 3, then that's like adding a word or deleting a word, which would have an effect but no longer be a frameshift mutation. If I delete a letter, notice that now the first letter of word 2 is now in word 1, and once again it alters the reading frame. None of these words make sense, and so therefore this was a very serious mutation. Now then, going to DNA as opposed to those letters in a word, a point mutation could change whether, so if something like CUU was changed to UUU, now instead of coding for leucine, that would code for phenylalanine. So a point mutation could change the amino acid. It doesn't have to, because notice that, for example, all 4 of these, CUU, CUC, CUA, CUG, all 4 of them code for leucine. So you could change one to another and still code for leucine. And so a point mutation, once again, is just changing one letter, and it could change the original sequence, which coded for the amino acids phenylalanine, lysine, glycine, proline, arginine, arginine, tryptophan. Here it changed this one to a serine, so it changed one amino acid. Let's say I add 2 nucleotides, all right? If I added 1 in this particular sequence, it makes a premature stop codon, which would end the protein. But now this would change every downstream codon after this addition. And notice that all of these then are different from the original sequence. If I were to delete a nucleotide, then once again, all of these could change. Now, maybe by dumb luck, a few stay the same. But once again, a frameshift mutation is a much more serious mutation than a point mutation.